I'm curious. I am curious. Curious
by nature.
Welcome to Curious by Nature. I'm Jessica Johnson from Newswise. Today I'm speaking with the University of Florida scientist Dr. Robert Ferl. He is 1 week back from going to outer space, and I've never spoken to anyone who has gone to outer space, so I was very excited to hear about this experience for him. We also spoke about the research that he did while he was in space, and then we talked about new and fascinating opportunities that now exist for researchers. So without any further ado, please introduce yourself.
My name is Rob Ferrell. I'm a professor at the University of Florida. I'm also the assistant vice president for research.
Tell me more about your work in general, what you do and what you have done.
So I'm a molecular biologist. I'm somebody that tries to understand primarily how organisms adapt to their environment. For the longest time, control of the environment was the one way in which we studied how genes were activated or inactivated. It became something that we could control and therefore use as a tool to understand gene expression. The more we understood gene expression, the more we understood that if we could find out how genes were responding, we could actually begin to understand how organisms are adapting to their environment. So it became more of an outward-facing scientific interest to understand what happens to in our case, plants when they're in a strange environment, a challenging environment. And so during the space shuttle era, plants coming back from space, some of the original experiments of biology in space were indicating that plants were undergoing some kind of stress during their flights to space. It became a fundamentally interesting question. How does biology respond to being in an environment that is truly novel, something for which, in our case, the plant has no evolutionary adaptation, preconditioning, or otherwise knowledge of how to be in space? I find it fundamentally intriguing. What's the limit of our life? Can our life live on? What gives us the idea that we can live on another world? What kind of thing got in our minds that said, sure, we've grown up here on Earth, but we're capable of living anywhere? In our case, plants are what we're using to understand those limits.
Mm-hmm. Yes, fascinating questions. And there are so many more, I imagine, and many questions that you're looking at. So one of those questions, I guess, happened when you went up in space recently. So tell me more about that.
Oh, sure. Happy to tell you about that. The ability for a scientist to go into space, for me, a plain old ordinary university scientist, to go to space, I think is sort of old, new part of what is becoming the commercial spaceflight ecosystem. There are now spaceflight providers that we can use to do studies that we haven't been able to before. And NASA is now funding scientists to begin to do their own studies while taking those rides.
Were you the first scientist, non-NASA astronaut to go into space?
In this particular program, yes. But remember, in the space shuttle era, there were a lot of people that were payload specialists. There were astronauts dedicated to particular payloads or particular areas of science. And so the concept of what would you call it, non-career astronauts being a part of missions is an old one. But what I am the first to be able to do right now is to be able to write a proposal to NASA, to the Flight Opportunities and Biological and Physical Sciences divisions at NASA, pose a scientific question, and with enough curiosity, gather the funding from NASA to go ahead and do this. So yeah, I'm the first to do that particular thing, but I'm certainly not the first scientist,
right,
in space or something like that.
Yeah, so thank you for clarifying that kind of, I guess, entry point, though, because I think that is very fascinating.
It is, and one of the very, I think, and I hate to overuse the word cool, but one of the very cool things that this does for us as space scientists is it cracks open the notion that we can study a place, study phenomenon that happened in a place without ever having been there. Imagine being somebody that studies the oceans and never having been on a boat. Imagine being a forester and never walking among the trees. So for us as space scientists, and for me as a space biologist, I've now been there. I've got a whole new appreciation for what that environment is like. I've got I've got a different way to see things. I've got a more informed way to conceptualize what happens to biology in space because my own biology has been there. I get to see that with a whole new lens, simply because I've had the opportunity to go there.
So there's so many things to ask you about, and like specifically about your biology. What was that like? I mean, first of all, how did you prepare? What did you need to do to be able to go into space?
So the New Shepard Blue Origin flight system is pretty darn well designed and straightforward. So the formal training with Blue Origin takes about 3 days beforehand, because it's a pretty straightforward flight. But that would be giving the wrong impression for how a scientist prepares to do this. We first wrote this proposal more than 7 years ago. We have been working on various parts of the technology that we are developing for use inside the New Shepard capsule for years. We have done many experiments and many experiences in zero-G aircraft, so I'm familiar with the weightless environment and how to sort of manipulate your body and understand what the limits are of moving around in zero-G. So I have that training behind me. Not everybody does, but I do, because a lot of our laboratory work has been experiential and has been on flight lines in the past.
Tell us about the specific experiment and what were you looking at? I don't know if you have any results yet or not.
No, it'll take actually a couple of months for all the results to come in. But the story goes something like this. Over the past decades, we've gotten really good at understanding what it is that characterizes an organism in space, usually on the space station now, and an organism on the ground. Mm-hmm. We've done, for example, the twin studies compared the Kelly brothers, one who was in space for a long time, one that was on the ground. We know a lot about plants that have grown and lived on the International Space Station compared to plants that are here on the ground, and same with animals and microbes and all kinds of things. But there has been almost zero opportunity to ask about the transition from being on the Earth to being in space. Mm-hmm. Essentially, all experiments get started after the vehicle is docked with the International Space Station. So we collect no data on the way up, but no data on the way down. So these suborbital flights give us the opportunity to characterize those first few minutes of going into space. High G, ascent phase, the microgravity of being in space, and then just before reentry, we can do the reverse and collect samples just before reentry and after we get down to the ground. So our big goal for this flight is to begin the process of characterizing those initial few minutes of entry into space. What do the gene expression profiles look like as, in our case, a plant enters into the spaceflight environment? And what happens when we return from spaceflight?
From what I've seen, correct me if I'm wrong or whichever part of this, so I'm just trying to understand, like you basically took these vials that were airtight and you activated them at different phases of the flight. The activation means that you just expose them to those circumstances. So, for example, some of the plants, they were exposed to when you were taking off, others while you were just up in space, and then others as you were coming down and landing. So that means, I guess, you expose them just at those particular moments, and then you're taking them back to the lab to see if they grow in normal conditions, or what are you looking at?
Yeah, so to get a little bit more deeply into that, fixation tubes, KFTs, Kennedy Space Center, fixation tubes are spaceflight qualified pieces of hardware. In other words, they can carry a preservative in one compartment and the biology in another compartment in a safe way inside a spaceflight vehicle. They've been used on the space station and the space shuttle for decades. Mm-hmm. Our technology development was to find profiles, procedures, and deal with all the safety issues of using them inside the New Shepard capsule. So the first part of what we had to do in the technology development phase was work with Blue Origin in order to make sure that we could take the KFTs. How many could we take? When could we activate them safely, and what parts of the flight profile? And once we worked all that stuff out, we designed the experiment so that, now I'm going to answer your question, at various stages of the flight, the plants were exposed to a preservative that keeps them biochemically frozen in that molecular state. And so we can look at the gene expression patterns that characterize the plants at that moment. So the plants that we took up experienced either just the ride to the launch tower, or the ride to the launch tower and the ride up to space, or the launch tower right up to space and microgravity, and then the return. So each of the plants experienced some part of the trip to space and back. And what we did was biochemically freeze them at each of those locations.
What do you do with them once you've brought them back to the laboratory? What are you looking at them to see, I guess, what their gene expressions are and if they're still living, if there's, or they were killed in the up and down or whatever.
They almost assuredly were not killed in the
Okay.
And all of our analyses were, we've got now high resolution photographs from them before and afterwards. So if, for example, they got busted up by the landing or something like that, we would know through photography. But our work involves, once those samples get home, is we basically grind them up and extract the RNA from the shoots and the roots and the leaves, and ask, of the 25,000 genes that are in each of the cells of a Ravidopsis, this plant, what genes were up, what genes were down during the various phases of the flight. In other words, what metabolic pathways was the plant activating in order to deal with that particular environment.
And the Ravidopsis that you said, what is that?
Ravidopsis is a wonderful little plant. It's been often used in spaceflight because it is small and can go through its entire life cycle in a fairly short time. But its genome has been well characterized and sequenced. It's used by thousands of scientists all around the Earth in various environmental conditions, and its genome is very well studied. So it is one of the what we call model organisms of the plant world, and allows us to compare our spaceflight data with data from all kinds of environmental exposures here on Earth.
And how has this experience changed your life, if it has at all?
Well, in the week after the flight, and has it changed my life? I think the answer is yes. I'm finally getting to sleep again, and I'm beginning to analyze my own personal experience in space. But I can tell you that being in space and seeing the blackness of space compared to the blue band of atmosphere around the Earth is something that does indeed, I think, change a person. I can tell you that during the time that I had to myself, I was totally absorbed with looking out the window. Being weightless was not such a big deal to me, but boy, looking out the window and experiencing my version of what is called the overview effect, the idea that you simply create a different perspective of the world and yourself by seeing it from that distance, that's all very real. And I'm still in the early phases of processing what that means to me personally, but I can tell you it's pretty darn profound.
Was there anything that was surprising to you through this experience and going to space, or your research in particular?
The scale of work that it took to make it happen was, in retrospect, a bit surprising because of the amount of work and the number of people involved in making my simple 15-minute flight possible is amazing. My thank you list of people that have been involved is incredibly long. The other thing that is not truly surprising, but surprising again in its intensity, is the feeling of being humbled by the experience, to be able to do something for which many others ought to be able to do. and should have been able to do, or something like that. So I feel actually pretty small in the world of, space adventure, but holy cow, I got to go. And, that's an interesting mix of emotions there. The other thing that I hope is not surprising at all is that when people see that it is possible to be in space, to go to space simply because you're curious about space, you want to study space, you can write a proposal and go. That means that there's a whole new group of people, scientists, curious people that can go to space simply because they want to study space. And for me, that's actually pretty profound. And one of the big things that I hope is a part of the success of this mission, more people going.
What does one do after you go to space? See, in your field, what are the next steps of your research? Are you thinking about that, or are you just trying to come back to Earth?
Right now, I got to tell you, a week after, I'm still trying to come back to Earth. No question about that. The profoundness of the experience is not to be underestimated. But what's the next thing? I want to make sure that the door stays open for more scientists to do this. A part of my job I now see is advocating for more people to get more science in space and to conduct the science themselves rather than handing them off to somebody else.
Do you think that people know about this? Do you think that researchers know? I mean, you were kind of in the space field, right? But many researchers who aren't really focused on space might also be interested in space. So I would imagine they might not know about it.
Exactly. And I mentioned that one of my perceived duties now is to be a strong advocate for this. I really want to see the program grow, and I want it to be something that attracts all kinds of scientists to the space field, not just because they get to go.
Right.
But if they do get to go, it does add a dimension of interest. Again, back to why would you study the sea if you were never going to go there? But now you can. You can go to space.
If you can finish this sentence, I'm curious about
the limits of life in the universe.
Thank you so much for speaking with me today.
Curious by Nature is a production of Newswise. More than 7,000 journalists get the Newswise wires. Visit newswise.com and find out how to spread your news today.